Let's get started. Eliezer asked me to give an extra talk to help fill out the schedule with one of the cancellations. So then I'm happy to do so. And in some ways, this is a nice opportunity because the projects that I'm going to speak about are a mixture of population genetics and microbiome work. So it maybe works well for the theme of this conference. The title of this I've put is Inference of Haplotype or Community Composition in Pooled Sequencing Data. So this project starts in the world of massively parallel sequencing, which Dan did a great job of kind of introducing us to some of the promise and perils that come with these technologies, that they give us a great throughput, but there are many challenges of working with this kind of data, such as the uncertainty in the read mapping, the high per base error rates, and the variable depth of coverage. So we always have to keep in mind that bases are sequenced imperfectly. So on the left panel here, you can see error probabilities versus the position on the read. And as you go along the read, the errors are more commonly occurring because of the way the sequencing is done. The images start to get out of alignment. The fluorescent signals become less clear. And because of that issue, when you look at these base quality scores that Dan also introduced us to, as a function of position on the read, they start quite high at the beginning of the read. And so these are histograms of the quality score distributions. So in the first panel, you see all the quality scores are quite large because the reads are very high quality. The base calls are very high quality. And then as you move down, say, to position 75, the quality scores have now moved over to be much lower, with many more of them getting a quality score of 2, which in the Illumina coding scheme basically means that there's no call for that base. Okay? And a principle here is that these base quality scores can help us model some of these complex processes, like how the errors increase along the read length. You know, we don't have to model that. We can let the base qualities capture some of that error. And so we are going to be using a principle shared with Dan's ideas of actually trying to keep track of these base qualities to help us improve our inferences. So, okay, so in life, cost constraints lead to difficult choices. Okay, and in our world, that means like sample size versus coverage, using indexed reads where you do some particular library preps that add a little barcode on each read so you can separate out individuals, versus just pooling the DNA and not being able to untangle which piece of DNA comes from which individual. And while a bunch of my lab's work is with like human DNA projects where there's lots of data or lots of budget and indexing reads is possible and you don't need to use pooled sequencing, there are lots of situations in non-model organisms, especially where people are looking for ways to cut costs. And in a way that comes to this point here, someone might say, why worry about these kinds of problems of like how to help people do pooled sequencing? Because those problems will go away. In another generation, the technology will be so cheap that no one will need to do this anymore. And but even as the technologies advance, the cost constraints still will not disappear. There's always people who are working with a small budget. And beyond that, like users will always be trying to push the limits of sample size. So they might be willing to, for a particular application, want a very large sample size, and for that reason want to save costs by doing pooling. And as I mentioned, especially in these non-model organism contexts where oftentimes the budgets can be quite small to study some relatively understudied organism. And then also, a reason to do this is sometimes it's just good to tackle the problem in front of you. Yes, the problem may go away, but we're sitting here in this generation, we want to solve a problem right now. And then it's fun actually because I was asked to do this talk relatively last minute, and the first version of this talk I gave several years ago. It's nice that, well, these problems haven't gone away. Yeah. And they've morphed. So right now, single cell sequencing makes thinking about low coverage data, ancient DNA, this stuff didn't exist when we started this project, and yet like the insights that we've gained might help in those settings when we didn't even expect them. So So that's some coaching on like thinking about, don't let people say, oh, experiments can solve this, you know. A lot of times that takes longer than people expect. Okay, so let's try to think about these problems. And we're going to do it first in this setting of what are called Evolve-And-Resequence studies, which is a type of study being done in the evolutionary biology community, especially in Drosophila. And the goal is to map the genetic basis of complex traits. So it's like GWAS in a way. And the idea is that you start with a set of founder lines that differ in a particular trait value. And so to make this concrete, the collaborator we work with has started a population from 162 founder lines of Drosophila that were fully inbred. So they're homozygous across their whole genomes, and they were fully sequenced. So we essentially have 162 haplotypes that then are put into a random mating population, and they mix. And then in this lab, they then pick a particular complex trait they're interested in mapping, select on it. So they take the top 10% of the individuals for that trait value, allow them to reproduce, then do that again generation after generation. So that's selection for extreme values of the trait. So maybe in one line of experiments, they select the, say, the largest individuals. In another line, they select the smallest individuals. And then at the end, they do pooled sequencing and try to compare either the allele or haplotype frequencies between the two pools. And the loci that have differentiated the most should be loci that are linked to QTL for the trait. Okay? So it's potentially a very cheap way to map complex traits, because all that's needed is to do these selection activities, which can be done with a set of technicians, and then one round of pooled sequencing at the end. Okay, and so let me show you then an example of this kind of work being done by our collaborator Tom Turner, trying to map a complex trait that's courtship song. So they're interested in saying we have a very complex trait. What is the genetic basis of how Drosophila sing? Okay, so you need to kind of break that into a particular measurable phenotype, and for them it's the interpulse interval. So that's like a little representation of their song with these little chirps, and there's a pulse interval between them that varies between species and between lines. And so they selected some high lines. So there's the interpulse interval in milliseconds for 2 high lines, 2 low lines over 14 generations of selection. They got a response in the phenotype that suggests that there's some genetic basis to it. And then they compare the allele frequencies in one long interpulse interval line versus another. So, and the interesting one, I guess, is the red ones that are short versus long. And the idea is that some of these alleles that changed a lot between the two conditions would be loci that are involved with this courtship song. Okay, and then by sort of combining these results, yeah, they were able to zero in on this syntrophin-like 1 gene and get a clue of the genetic basis of such a complex trait. So it's very cool. It's an emerging area of complex trait genetics. There's a lot of papers going on in this area with several groups kind of investing quite a bit in it. So the question is, how do we best analyze such data? And the vanilla approach would be to just have your read alignments from the pooled sequencing, and then go down each column and count the number of bases, the number of reads supporting one base versus another, and use those as frequency estimates. And that's what Tom and his group were doing, as others were in this field. And I heard about this. That given that you have known founder haplotypes, you have inherent LD in this because it's a small number of generations of free recombination after the founding of these populations, that the reads themselves even oftentimes cover multiple variant sites, particularly in Drosophila, who have a, remember yesterday I was saying humans have a pretty low level of diversity. We're 1 in 1,000. Drosophila have 1 in 100 differences per base pair. So what that means is on a single sequencing read, you can actually have, you might hit 2 sites that vary. So there's some haplotype information even in the reads. And then you have all this useful information in the base quality score, so you should be able to do a lot better than just counting alleles in the pileup files. So our goal was to produce a base quality aware approach that can infer haplotype frequencies. And as a schematic of this problem, what I'm showing is up top the known haplotypes, with each color representing a different haplotype, the dots representing different variant states at like polymorphic sites. And then you have some true haplotype frequencies in the final sample that you subject to pooled sequencing. And you get a set of reads, and you're hoping to get out estimated haplotype frequencies. And the challenge is that for each read, it might be ambiguous as to what its identity is. So that's why those reads there have two colors, like the green and the orange, for all the reads that could be equally mapped to the haplotype with the green or the orange. Okay? So there's a key latent variable here, which is the haplotype identity of each read. Because if you told me that, that one that's green and orange was actually green, or actually told me all of them, then I could just count the number of reads supporting each haplotype and thereby get an estimator of the haplotype frequencies, right? So, so with that idea, there's this key latent variable. We can kind of add that to a model and build up a graph for this scenario where we have underlying haplotype frequencies in F, then that generate a pool of reads that have haplotype identities. And so that's going to be a multinomial distribution for the read identities, really the counts of the read identities. Each read is like a multinomial draw from a frequency vector F. And then when you have that haplotype identity, then that given the base qualities and the matrix of the actual founder haplotypes will allow you to write a distribution on the observed base in a particular read from the experiment. And so that's represented here, where if Q is the base quality estimated error probability, then you have a exact match with a probability of not having an error. And then if you do have an error, then we're just going to say that you will have one of the three other bases. That is, one of the three other bases than the one that's actually the one of the haplotype founder, the founder haplotype. Okay, so this is a relatively straightforward model to work with. It looks a lot like the latent Dirichlet model and many other kind of simple latent variable models. And so there's an EM algorithm to solve it. And so it's this iterative updates of the allele frequencies over EM iterations. And so that's the formula there on the top for the frequency update in an EM step. And that first line where you see that it's an average of these expectations of an indicator variable is indicating that what's really happening here is that we're probabilistically assigning reads to haplotypes. Okay, so that's the first step, which we're not going to take like a best match, like string match, and assign it definitively. We're going to have these expectations of indicators, so they're probabilities, and so we're going to put a mass on each founder haplotype proportional to the probability of the read coming from that. Okay, so you can think of it as like a soft matching. But when you expand out this expectation, I also want to show you that it kind of has two parts. One part is this product over the sites in the read that's based on the base qualities. So this is the part that incorporates the base qualities. So that should be helpful instead of doing like naive, like, oh, well, I'll accept one mismatch. Well, here we're like, it depends on how good the base quality is, whether we're going to accept that one mismatch, right? And then here's another key part that comes out of that expectation. The PZ equals K given F from the previous round. So the algorithm is sort of learning the frequencies in the pool and then using that to help shape what it thinks about the identity of any one read. So it's pooling information across the whole sample. Once you've already gone a couple iterations in, you might have learned that 80% of the haplotypes are from, 80% of the reads are from one haplotype. So now you get to a read that you're kind of uncertain whether it belongs to that haplotype or not. Because you know the pool's really enriched for that haplotype, you're going to push that read in that direction, right? So that's another way in which this is smarter than just doing like a one-by-one exact matching, because you're sort of taking into account, well, I'm pretty sure from all the other reads that this is the most abundant haplotype, so I should push any uncertain read in that direction, and that should clean up my inferences.
Good. So the probability is not by exact matching, rather the probability of belonging to one haplotype is not by exact matching, rather you allow the error to be along the read.
Yeah. Yeah, so we're assuming each position in the read is independent, and you either have an exact match with probably 1 minus q, or q is from the base qualities, or a mistake. Yeah. Yeah. Okay. So these properties are properties of an EM, in this latent variable situation. I want to highlight just qualitatively some of the advantages of working, this kind of framework. And so that shows up here in how we perform when we compare to other methods. So the y-axis here is the sum of squared errors for the haplotype frequencies that are estimated. And I'm getting a little, sorry, I haven't looked at these slides in a while. We're comparing to an allele frequency-based method. So what I might be showing is a sum of squared errors for the allele frequencies, because if you have the haplotype frequencies, you can collapse them to get allele frequencies. Okay, so it might be nice to compare against, since like our collaborators, the first thing that we're doing is just estimating allele frequencies. We can say, well, we're going to estimate the haplotype frequencies, collapse them to allele frequencies, and they should be better allele frequencies than you started with, right? So, okay, so Hippo in the purple is this allele frequency-based method that doesn't use any haplotype information. Then in yellow is string match. You just assign each read to the founder haplotype if it's a perfect match, otherwise you throw it out. Soft string matches, you assign each read to each haplotype based on its probability of coming from that haplotype, but you don't actually do that EM that gets the pooled information, pools information. And so you can see how then when you do the EM, you do much better than any of these other simpler approaches. Okay, so that's exciting. Okay, then factors affecting the performance are kind of obvious. If you increase coverage, you do better. You increase your read length, you do better. If the reads are more error prone, you do worse. So we investigated all these things. And we also investigated the effect of base quality score calibration. So this is reiterating a theme that Dan brought up, is if you're going to use base qualities, which would seem smart to do, remember that motivating slide at the beginning, they capture how like the ends of the reads are much worse than the beginning. It should be much better, but at the same time, these base qualities are estimates themselves. So if we just take them straight off the machine, they can bias us. And so doing some kind of recalibration approach can improve the performance. So this is the kind of curve we had with a similar decay for the high quality values that Dan emphasized, how high quality things are actually typically not as that high of a quality. For these particular libraries, actually, it seemed like the low quality stuff was a little bit they're underreporting the quality. And then this is showing with no recalibration versus the recalibration, we do better. Okay. And we can ask how this works for improving power for the Evolve-And-Resequence. And so if you use these haplotype frequency estimates, you do better than using the naive allele frequency-based estimates. And let me keep moving along here so we get to the microbiome stuff. And then this is a plot. This is kind of a fun one that Darren put together, where what you're seeing with these colors is the frequencies of each of the 162 founder haplotypes as you slide along one of the chromosomes of the genome of the Drosophila that were used in this experiment. And you see as you move towards this side, you get these huge enrichment of these two founder haplotypes. So this may be that there was sort of a soft sweep where the allele affecting interpulse interval existed on these two haplotypes and then got strongly enriched over the experiment. You'll see one of the big drawbacks of this Evolve-And-Resequence type of work, that if you're not careful, you get huge mapping intervals, right? Like in GWAS, we're used to getting things at the scale of like 100 kb. Here's like, okay, you've narrowed it down maybe to 10 megabases, still some work to be done. So, and in fact, Darren actually did a bunch of work in his PhD to ask how do you design these experiments as best as possible. Okay, so now let's consider a related problem, which is inference of microbiome community composition. So let's just think about a problem we were just talking about, and now this one. You're given short sequence reads from a microbiome sample. You've got reference reads from known taxa, like in particular like 16S sequences. So you have like a library of known haplotypes, the known taxa with their 16S sequences. You have a pool of reads that you come from that set of founder haplotypes, and you want to infer the relative abundance of the taxa. So it's completely structurally the same problem, okay? Well, we're going to be applying this to approaches where they've used PCRs to get just the 16S, yeah. And we are, to be fair, like ignoring here the unknown, that there will be reads from taxa that are not in the reference. Okay, so some people in the microbiome, they call this the closed reference problem, where it's maybe like for the gut, it's so well studied that most of the taxa are known. So, yeah, okay. So, yeah, you're interested in getting the frequencies of the different taxa within a sample as opposed to the frequency of different haplotypes. You're using 16S ribosomal DNA, where you might be looking at the whole thing or just PCRing up just one part of it. And a lot of the existing approaches do this kind of hard match, like is it within 97% identity, then count it as coming from that particular OTU. But with the kind of non-trivial error rates of read data, you might think you can do better. So it's a case where it's a different problem, but it has the same graph, okay? And really the thing that has to change is just your interpretation that now these Z's are not haplotype identities of reads, they're taxon identities of where that read came from. And we first did this by applying the software from our first approach directly. And we could see that the accuracy improved, but the computational cost was really high because our first problem was sort of geared towards working with like 162 founder haplotypes. But in the microbiome world, you might be considering a reference database that has thousands or tens of thousands of potential source taxa. And so one of the things you need to do this EM is the probability of each read coming from each reference. So that's a very large table that becomes really unwieldy and slow. So Mark Rappel, a postdoc working with me, greatly focused a lot on how to improve the speed of that algorithm. And what he was able to do is merge a Kamer-based approach to quickly narrowing down the possible source taxa for a read onto the front end of this pipeline so that we could very quickly run sort of all reads against all taxa. Okay, so if you've heard of Callisto that Lior Pachter developed, that approach is what we're using on the front end to come up with a set of possible source taxa that then we then consider each query read against that small set of sources and do an exact local realignment, consider the base qualities, and then do the EM algorithm to get these proportions. Callisto does sort of the first step and then jumps right to the fourth in that it tries to get reads that have a clear taxon match and then does, a small set, and then does the EM directly on that without this local realignment and considering the base qualities. So, so they're related algorithms, but but we have a little more sensitivity because of that. Okay, so how does this work? We've done some simulations, and it performs quite well. So these are going to be 1,000 taxa with different Shannon diversity values of the communities that we're looking at. So across a range of different diversities of the communities, this is like the per reference, like how you do it, calling each of those 1,000 taxa, or if you lump and ask, like, how well do you do at the genus level? And this is a comparison to a bunch of existing methods for community composition estimation. And in general, we do quite well. So the blue line is CARP, and then the brown line is a collapsed version that we have for dealing with the fact that many references may be similar to one another. And yeah, so in general, we do quite well. Maybe the most interesting things to say are that Callisto doesn't do as well with the short, just like the V4 hypervariable region. And then if you look at different read lengths and you have very long reads, Callisto also doesn't do as well because there's so many differences within a long read that it actually it's sort of too stringent in calling a match. If it sees too many differences, it won't use that read. And so you end up using many less reads than you actually have in the sample, and you end up getting noisier estimates overall. So we're good for accuracy, but we take a hit in terms of computation time. So Callisto is sort of the third row, is probably the most relevant comparison. You can see CARP and our collapsed version of CARP quite a bit more expensive, okay? My Monty Slatkin, my wise PhD advisor, used to say, you know, if an experimentalist has spent months collecting the data, they can afford to run it for a couple days or, you know, a couple hours. So I'm going to pull that line out of the hat for this one. Okay, so this has just been a fun problem. That's sort of a mix of a PopGen problem that then the structure of the problem is the same, but in a different genomics context. And to summarize, yeah, there are these advantages to modeling base errors, using these base quality scores, pooling information instead of doing sort of treating each read at a time, like thinking about the whole inference problem jointly. And yeah, and as I said, where we can repurpose these tools. So these are the readings from this work by my former PhD student, Darren Kessner, who was a bioinformatics PhD student here, one of the first, in one of the first cohorts. The first, yeah. And then Mark Rappel, my former postdoctoral fellow. HARP is the piece of software for haplotype analysis of reads and pools. And then its riff here is CARP for K-mer-based analysis of read pools. I want to finish with two quotes, which is, this is from a quote I found, a really nice blog post on computing costs and sequencing. So I'm nervous that the new era will really be one in which the net rate of discovery is finally not limited by sequencing technology, but by available computing capacity. A nice quote to remind us of the importance of what we're doing here, of thinking about, you know, these computing challenges. And then this other quote by James Crow, a really foundational statistical geneticist and population geneticist. We'll see with a little ingenuity, we can do a great deal of genetic analysis of natural populations, despite our ability to carry out controlled experiments. So both, I think, in population genetics and in microbial work, it's very hard to do experiments. But it's sort of the cleverness of people in this room that kind of allows us to do good biology. So that little bit of ingenuity is what we're aiming for. Okay, and I want to acknowledge Darren, who's there from a lab retreat in the sunny, happy LA days. And then this is Mark Rappel from the wintry lab ice fishing trip. This is Joe Marcus. This is not DiCaprio from The Revenant. This is Joe Marcus. And then Tom, who is a collaborator on the Drosophila work, and Katie, Simon, and Jack, who helped give advice on the microbiome work. And then thank you guys for your attention. So. Oh, yeah. That's a great question. So, the question is about modeling recombination. So, the way that we frame the first problem is actually, we're doing the haplotype frequency inference in Windows. So, sort of just looking at reads within, say, a 10 KB window. What are the representation of the source haplotypes? That is probably not so bad because it's only, say, like 15 generations of this selection experiment. And so you don't expect too many recombinations to occur within any small window. Yeah, so that's how we tackled the problem. It would be attractive to think about some sort of, HMM type model on the that kind of thinks about recombination along the chromosome. I think that would be a lot more engineering and machinery, and I'm not for probably some small gains, but that's a great point. And then yeah, for the 16S, you're intuiting it right. It's such a small piece of DNA, it's probably fine to ignore, but I'd leave that to the microbiome community to sort of think about that, yeah, or comment on that.
I was going to ask you
Yes, we have, and it's on trying to use like monomorphic sites. Yeah, it's a little trickier though, right? Yeah, so yeah, but I can't recall sort of the impact of that. Yeah, but it is definitely something to think about. Yeah, I suppose you can always run these by flattening the base qualities to be uniform, and then it would be equivalent to just ignoring the base qualities altogether. So if you were so nervous about the base quality calibration, you could say, well, let's just remove that part of the modeling. Yeah. Yeah. Okay, great. Yeah. Yep.
Okay, so now we're going to close CGSI. You have to get it up?
Yeah, I think I can get it to. You want me to try? Yeah, let's see. Yeah, I'm just going to see it. Yeah, it's not automatically advancing, so I will be the advancer. Okay. It's projecting. Yeah.
Yeah. So we have, okay, so while we were doing this, we have back in case. First of all, a lot of people to thank. First of all, can all the volunteers stand up? Okay. So these are the people.
Sorry.
Huh?
Do you want me to keep advancing, or should we wait till the end? Okay. Okay.
Yeah. So,
It's going to distract.
This is low, thank you for so these guys, these are the only ones that are left because the other ones are tired from weeks of this, but they really, we had a probably group of like 16 or so. They really did everything from getting breakfast to everything that went through the program. They did everything. They also are really probably just working with their faculty at UCLA because they're wondering why so let's thank them all. And then the other thing is that this year is quite a different year than previous years because we actually are one real major goal of CPSI is to democratize this, turn this more into a community rather than kind of a UCLA-oriented thing and program. And so this year what we did was we actually have an organizing group who we recruited, and they kind of agreed. And they didn't, I think that they certainly did not realize how much work went into this. And then probably every Wednesday at noon for it seemed like multiple years, even though it was only probably 6 months, we met. And then when they got here, I don't think they really realized that they basically have slave labor conditions from breakfast, being time for breakfast through 1 in the morning Hollywood Bowl type activity. But we're really grateful. So if I can have Ron, David, Kirk to come up. For you guys. So Faradun had to leave, and but Faradun and David actually organized the first week, and Ron and Kirk obviously this week, and just huge amount of work and help until we got to it. So these are for you guys. Thank you. Yeah, there you go. Cool, great. So I think that, did you go through all the photos?
No, still going.
One of me.
Yeah. Going diving for it, yeah.
That was like a glorious
Hanging off the net, catching your breath, yeah.
Yeah, that
Nice.
was fun. Yeah, there's a lot of injuries.
Swarthy.
This is before your toe was broken, right? Yeah. Maybe the moment up. Yeah, maybe. Yeah. Nice.
Nice.
And also, I want to say that, you know, look, we, for faculty, all the faculty here, you know, we ask people to come here for a week. And we're pretty strict about it. And some come for 2 weeks. Some come for 2 months or 2 weeks. But you know, that's a lot of time. And we also ask all the faculty to go to everything, and we don't tell them that until they're here. So like usually 8 in the morning, the first day, we're like, oh, you have to come to this and this and this, and they all agree. So it's great. It's a lot of work on them, and we appreciate that. And then also, you know, we've had many of you have been here for a month, and that's also a large position. That's a big chunk of time, and we thank your PIs for letting you come here. That's also too. That really, really great conference, and you know, and for me personally, you know, lots of all the time. But yeah, it looks like you guys had fun.
And maybe I'll speak for all of us, and let's thank Eleazar for chief organizing. And Iran Halpert, yes. Great.
So as soon as John finishes.
Okay, I'll pick it up. There are some great photos here. My gosh, yeah.
That's what the house is like. I've never been.
Wow. These like the iPhone 8, like the fuzz.
I think that these are some souls in the app, right? Is that what it is? You know, that's the giant screen.
This last set is just generative adversarial network shots. Oh, Getty, yeah. Oh, yeah.
Please add a soundtrack.
I think we made it. That's it. All right.